Hello, in this lab, I'll be briefly going over how to analyze data for the Compaction Lab. The expected result of analyzing the data is to produce a graph similar to the one shown on the left. For your lab report, you'll have to add an extra step and calculate the dry unit weight of the soil and make sure of the units. Separate your data between the modified proctor and the standard proctor. If there are data points that are outliers, you should remove them from the analysis and include them in the report and consider why there is a difference. Finally, calculate the optimum water content and the maximum dry unit weight of each of the two proctor tests. The lab data you have should be similar to this table. The values for this table are for demonstration purposes only. The SP and MP column is for determining whether the data was a standard proctor test or a modified proctor test. The target water content was for determining how much water was to be added to the soil for mixing before compaction and could be compared to the actual water content. The first three weights or masses for water content calculations, and the last two are for density calculations. Because the lab was conducted using a standard compaction mold, we can assume the density to be 1/30th of a cubic foot. We will need to calculate the actual water content, mass of the soil in the mold, and both wet and dry densities. Here are the three formulas needed to calculate the dry density of the soil. Using these three formulas, we can calculate that the dry density of the data from the previous slide is 1.43 grams per cubic centimeter. Note that the volume of the mold is given in cubic feet and must be converted. For the end result, calculate the dry unit weight from the dry density and plot all the points on a graph. Determine the maximum dry unit weight and optimum water content for both curves.